So if you're just joining us now, we are at the GNSI virtual conference. We have our next speaker in the session on creativity in the time of COVID. This is Jennifer Fairman. We're so excited to have you. Jennifer is an associate professor at the Johns Hopkins School of Medicine. She's a widely published medical illustrator with publications in or illustrations in almost over 960 books, journals, websites, videos, etc. She's an accomplished businesswoman. She just finished a Master's of Professional Studies in the Business of Art and Design from the Maryland Institute College of Art, with perfect achievement, whatever that means. Who does that, Jenny? We're delighted to have her here with us today to talk about the challenges of illustrating an evolving story with COVID. And Jenny, when you are ready, we are ready to pull up your slideshow. Thank you so much for being here. You're insanely busy.
Yeah, it's been busy. Sorry about that little. Just when I'm about to start, my daughter knocks on the door. So of course,
classic.
I had to kick her out of the cockpit, right? So sorry.
No worries. Well, I'll let you have the stage.
All right. Thank you so much. Thank you so much. I just want to thank Cappy personally for helping everybody, including myself, get tech savvy here today, and of course Sarah and the entire GNSI for inviting me to speak to you today. Let's get started. I have let's see here. I'm going to share my screen. I'm going to share. And thank you all for the great comments in the corner. It's so great to see so many names, and I'm sorry we can't see each other in person, but hopefully, someday soon, of course. I'm going to be talking to you about COVID-19 and visualizing the SARS-CoV-2 virus itself. Here are my disclosures. And before I begin, I'll just warn you, I'm going to begin by reading something that I wrote a couple weeks back. And it's a thought that's been in my mind ever since this whole pandemic started. So bear with me, it's a little heavy, but it kind of frames what I want to talk about. So I can remember listening to the TV early in the morning of September 11th, 2001, and we all know what happened. The chaos, the questions, the surprise, the unknown future to come. Back then, I was running my small studio practice out of a rented office space, which was walking distance from my apartment. And I didn't know the full impact of what was happening at that moment. With projects on my mind, I turned the TV off too soon and was focused on getting to work early and in a hurry. When I got there, I tuned to NPR. That's when reality began to sink in. I sat focused on impending deadlines, but suddenly realized that none of it mattered. Hundreds of people were dying as I sat there. In that instant, my role as a medical illustrator was completely irrelevant. Deep shame came over me, and I felt completely worthless to those who were suffering at the moment. I'm just a useless artist. How can I help? I don't think anyone enters our profession ever thinking that they might feel that way about their choice to become a science communicator. There was a time much earlier than college that I had aspired to become a pediatrician, a useful health professional. And on 9/11, I felt helpless and deep regret that I had made the wrong choice. Though the US was experiencing a targeted crisis, the world felt it. People came together, put aside their differences, and helped in every way possible. Strangers greeted each other with extra kindness as many felt isolated and helpless. This was what would become one of those times the world came together to fight an elusive killer. Now we are facing a similar moment. When the first murmurs hit the media that a novel coronavirus had been discovered in an open market in Wuhan, we knew we might be facing yet another elusive killer. The questions began to flood. Who was patient zero? What host did it originate from? How fast does this thing mutate? How does it compare to influenza? What are the best ways to prevent the spread? What makes one patient more susceptible than another? Was it carried into the Wuhan market from a nearby farm? Will we ever develop an effective vaccine? Will the world go back to normal? With a boundless global economy, it was inevitable that it would make its way into everyone's backyard. Epidemic became pandemic. Mortality continues as we speak, and as researchers frantically work to find answers, effective treatments, and a cure. Our field is seeing a surge where we can't draw pictures fast enough. Indeed, there have been so many that I personally have lost count. Our best weapon is rapid, widespread, effective communication of trusted, factual information and discrediting polarized and politicized misinformation in a fragmented media environment. Yes, the world is in crisis again, with one major difference. I am needed. We are needed. So now we have this great responsibility. Back in February, one of my colleagues, Michael Konemus of Emory University in Atlanta, posted on the Association of Medical Illustrators Hub Forum. He says the following: we have been working on illustrating the SARS-CoV-2 virus, and it seems to be transitioning into a very dangerous pandemic. We in the AMI are uniquely positioned to help share very accurate information with the public. I would like to get a virtual group together online where people will be able to share the latest accurate information related to what we might illustrate about this virus. With COVID-19, I believe we have a responsibility to be more collaborative than competitive. And I couldn't agree more. With this admirable statement, the COVID-19 Medical Illustrators Forum was formed and began compiling what is now a nearly 70-page resource by 21 plus contributors called the SARS-CoV-2 COVID-19 Research for the Medical Illustrator. And it's a shared Google Doc. And this is the handbook that I would refer to and contribute to for every COVID-related project since. Suddenly, we have what we call, or what I like to refer to as this COVID colloquialism that now everybody's getting used to seeing in everyday speech. Novel coronavirus, social distancing, flattening the curve, contact tracing, all this stuff, right? This is where our journey begins. And when fear of the unknown begins to spread, it's critical to pause and research and educate. Who is the enemy? Science communicated through art leads to understanding. Understanding leads to better choices and hence better outcomes. So enter the most seen and disseminated visual of SARS-CoV-2, the CDC's didactic, fiery, gray and red spiky ball created by Alyssa Eckert and Dan Higgins. These two explain very nicely creating visual representations of diseases provides a way to take something complex and abstract and make it tangible through visualization. One goal is that through this visualization, we can help inform and educate the public about the disease, and thus hopefully help improve public health by limiting and halting the spread of this disease. So, and again, this is such an early on moment for me. You know, back in February, March, I'm starting to get emails and calls from various clients. One of them who was a microbiologist and immunologist and works in infectious disease. And he's submitting a paper to a journal and wanted to talk about creating visuals that communicated the spread and prevention of various infectious microbes. And he's saying to me, this is going to be a big deal. You probably heard the news. There's going to be something like a tidal wave, you know, headed our way. What was coming exactly? And this is a figure that this is the figure that he called me about to kind of explain various microbes and what where they are on the scale of susceptibility to microbicides. And you can see SARS-CoV-2 right here, circled in pink. It's just this tiny little name in this long laundry list of all these different infectious diseases and microbes. And it didn't really, it didn't quite click yet. I didn't fully comprehend the storm ahead, but this is one of the things that was helpful in me, just kind of understanding where it lies in terms of all the other things that are out there. And then next I get a call from a friend who's up in Boston, and he says, back in January, he was worrying about COVID-19 and started an effort to produce N95 respirators, which, by the way, I didn't realize that a mask like this is actually considered a respirator. So there's that. And he needed an illustration for a product that his company was basically stopping the presses of what they normally do and creating this to help with the shortage. And you know what happened? Things accelerated. There were mask shortages, gloves, hand sanitizer flying off the shelves. Toilet paper became scarce. Who would have known, right? And this is a snapshot of where we were 6 months ago, which is crazy. So at this point, this is what the world looked like. And then came the big call. And I usually get one of these, once every year, once every 2 years. But across the street from where I work at Johns Hopkins is the Bloomberg School of Public Health. And they called to tell me that they had a project that I might be interested in. Basically, Bloomberg was looking to put together a special issue of their quarterly magazine, and it was going to be devoted completely to COVID. The summer issue, which actually just came out in July, and you can actually see the entire issue online. I'll show you at the end of the talk. But I'm looking at this, and I meet with them, and we kind of have this discussion. And I kind of sat around the table with the team, with the editor and the writers, and I said to them, "So what are we doing?" And they said, "We're going to look to you to kind of create the story." They didn't have a story written. So they kind of gave me this challenge of coming up with something that would explain to the public, something that would, be the narrative of, what is this virus? What does it do? How does it infect the body? What is the life cycle? And I didn't really think about the drug targets that virologists were, looking at it at the moment. Until I spoke to Andy Peacock, who was going to be the content expert for this particular article. Andy is the co-director of the Johns Hopkins Center of Excellence in Influenza Research and Surveillance, and is a professor of molecular microbiology and immunology at the Bloomberg School of Public Health. And his voice would essentially narrate this story. The day before, when I was meeting with my team, the day before, I was told that he was on Capitol Hill with other Johns Hopkins University experts in public health, infectious disease, and emergency preparedness. And they were offering Capitol Hill officials a briefing, officials who were seeking facts and perspective on COVID-19 and the new coronavirus as it began to spread. Congressional staff and media heard from Andy and his fellow researchers who had been on the forefront, essentially, of the international response to COVID-19 and advising on the best ways to mobilize resources and improve care and save lives. So from a virologist's perspective, what they're looking for are targets. What he's doing in his laboratory is he's looking at targets. So, of course, I had so many questions. I hadn't met with him yet, but I was preparing to, and I didn't know where to start. I didn't know, I started to think about all the stuff I had learned so far, and basically thinking to myself, well, what hasn't already been drawn that hasn't been covered by the CDC so well? Because the medical illustrators that I know, they're doing such a phenomenal job of educating the public. And I'm also thinking to myself, what's going to be relevant by July? So basically, to understand this thing, let's just start with the basics. So I'm just going to talk a little bit about the process that I went through in order to get to the story that would be eventually told. And in order to get there, you have to understand that, what is a virus? So essentially a virus, they come in many shapes and sizes and in different forms. Some are very complex, like a bacteriophage. Some are enveloped or have a lipid layer around them, like HIV or flu or even COVID. Some are helical shaped and they're enveloped as well, like Ebola. It's got a very unique structure. And then some have a very beautiful geometry, polyhedral, icosahedral geometry like adenovirus. But if we kind of drill down further, there's two types of viruses. So what makes a virus is that it has a capsid and also a genome of either DNA or RNA. But there are two types. There's either an enveloped or a non-enveloped virus. And you can see kind of the laundry list of all those infectious viruses that we were looking at before. Now we're seeing which ones are enveloped and which ones aren't. But of course, that's way too abstract for people to understand. And so I kind of evolved my infographic into something that looked a little more representative of reality, even though I still wanted to keep it simple and cartoonish. So this is what SARS-CoV is. I mean, it's essentially a virus that's enveloped. It has RNA in it, which codes for various proteins, various non-structural proteins, which you won't see here. They're only expressed inside the host cell, but then there are the structural ones. The most infamous that we see all the time is that red spike, or in this case, mine is green. And then the membrane protein, the envelope, which is a viroporin, it's like a pore. And then the nucleocapsid proteins, which surround the RNA. This is another beautiful representation, very simplified, so that people can understand what really makes up the basic form of the virus. Now here's the other thing. What is its relationship to all the other coronaviruses? So here we're kind of seeing the phylogenetic tree, and I don't expect you to even be able to read any of this stuff on the screen. I've simplified it for you in my next slide, but here's the Wuhan virus right here. It says WH human, that's what they call it. But then later is referred to SARS-CoV-2. And this is basically what you're seeing. So it's related to Middle Eastern, oh geez, I'm blanking out, respiratory syndrome. And then of course, SARS-CoV, which is here, which is mutated from a bat, and then SARS-CoV-2, which we don't know where it came from. So, but essentially what it does is it binds with particular affinity to the ACE2, it's an enzyme, angiotensin converting enzyme 2, on ciliated epithelial cells, which are found throughout the body, but especially in the lungs. It's found in the bladder, it's found in the GI tract, and a couple of other proteins, which I'll get into in a moment. But what it does is it binds to that in humans with super glue affinity. And in my conversation with Andy, he shared some good news. So SARS-CoV-2, interestingly, as an RNA virus, it has a very low mutation rate. That's good news. A non-structural protein, NSP2, acts as a proofreader. So there's all these different non-structural proteins that get encoded once it's inside the host cell. And he was saying that there's a lot to be said about. There's 16 of them, but this one in particular, he gave as an example that if it was knocked out of the genome, essentially because it's a proofreader, if it's not there, potentially, theoretically, the virus can mutate itself to death. So there's one point of weakness that we're already talking about. And the other thing is because the mutations are so low in prevalence, it's easy for researchers to figure out the genetic epidemiology, making it spread easy to track within transmission chains. So we're not just talking about contact tracing, we're talking about the transmission between herds. So from China to Iran to Italy to Boston, very interesting stuff. This is essentially the genome here, and it's separated into two layers. So the top layer are mostly where we see a lot of the structural proteins here, along with open reading frame polyproteins that get cleaved, and also, or no, I'm sorry, those act later on. But there's also the non-structural proteins that once cleavage takes place, these are all the non-structural proteins that can come of that, and again, get coded within the host cell and help the virus to replicate. So let's just talk about some of the basic the basic structural proteins. So first, as I mentioned, is the spike protein, which is this protein right here. Oops, it keeps going backwards. I'm sorry about that. So the spike protein, it gets clipped. There's two parts to it. There's S1 and S2. And S1, once it comes off, once it binds to ACE2, it gets cleaved and is able to then open into this pre-hairpin state, and then can basically what's happening. What you're seeing here is both the virus and the cell membrane of the host cell being pulled together like a tether. So this spike protein opens, and then it gets cleaved, and then it springs out and it pulls the membranes together. And then, there's like protein pump action that occurs, and then endocytosis, and it gets in, and RNA is inside the cell. Here's another beautiful illustration by Veronica Falconieri-Hayes, who also kind of illustrates this phenomenon. And I'm sorry, I know Gloria, you're watching. This is Gloria's beautiful illustration as well. So, there's a lot here. There's the E protein. Now, I don't expect you to read any of the stuff on this slide, but what I'm showing you right here is this is typically what I do when I'm doing a molecular illustration. I like to create what's called Jenny's cheat sheet. So I basically want to put every single thing I can that I want to remember about certain things, and also the various resources that I found them from. I want to make sure that I'm crediting and citing my sources. And so I'm putting all the molecules involved that are important to my story on one piece of paper, basically. So there's the spike protein. You can see it closed and open. I don't show the hairpin conformation, but we have the E protein again, which is a pentamer and is a pore. So we're seeing the top view here and the side view here. We have the nucleocapsid protein, which surrounds the RNA, and then the M protein, which I have to say is the most mysterious of all the proteins, because literally, as I was doing this research, there were people reporting in the COVID group, oh, I found this structure here. Oh, no, it's not on the protein data bank, but it's in the Zhang lab, or it's in the Fine lab, or it's in, all these different Swiss Pro, all these different places. But what's essentially interesting about this is these proteins, and I will admit, this is an artistic representation of what I knew at the time that I created that might not be completely accurate either. So this is my best guess at the time of what I learned. I understand that M is the most plentiful. The purple protein is the most plentiful. So this membrane protein, it's at an 8:4:1 ratio with both, let's see, so for every 8 dimers of M, there's there's 4 spike trimers and then 1 nucleocapsid protein. So, and then also E is like the one that's the fewest and far between. So this kind of gives you a sense of the structure of the virus and the role that these proteins play. And in addition, there are a few other proteins that end up being transcribed and translated within the host cell itself to help the virus replicate and assemble. So let's just talk a little bit about the geometry. I mentioned a little bit about this, but so this virus is essentially, according to research, approximately 84 to 92 nanometers in diameter without the spike protein in that calculation. It can be spherical or ellipsoidal in shape. I mentioned this ratio before. There's about 74 spikes per average size coronavirus, which is many more than I had thought in my head at the time. There's about 1,100 of the M2 molecules. So you can see there's a lot of those, and that's what contributes to the spherical or ellipsoidal shape. So we have these ratios here. And also the genome itself is approximately 30,000 nucleotides. So here I'm going to show you a couple of representations then by some of my colleagues in this COVID group. So here we have Melanie Connolly's glycosylated spike models that she created. And she was explaining how much she learned from a webinar that we all attended from Romy Amaro. And she explained the spike protein in probably the greatest detail I've ever heard in my life. How it doesn't just kind of plug into the virus, but it kind of sticks out using this little stalk here. Here's a beautiful representation by our colleague, David Goodsell. And it was interesting to read his description of what he was thinking at the time and where he was looking to the research and kind of, coming up with his representation. Here we see it surrounded by mucus and the cells that produce mucus and some antibodies. So we're seeing the virus kind of in its moment of an inflammatory response by the body. And then I thought this was interesting. This is another representation by isoform. Thanks to Nick Klein for allowing me to show this. When I saw this, I thought, oh my goodness, like this vicious virus is so beautiful. Like I want to wear this as a brooch or something. I mean, it's just the way they represent it. The geometry is just simply breathtaking in a way. And Nick Woolrich, he's been coming up with some 3D simulations here, showing both the outside. This is a great representation. Again, you can kind of see how, even though a lot of people are still trying to figure out what the stock is actually composed of, this is a very good representation of what it looks like, and how the RNA is packed inside of the virus. So this is kind of the process that I went through in order to figure out, how to create this infographic. And now I'm going to kind of fast forward through a lot of different visuals because we're going to get to the final outcome here in a moment. But essentially, this virus, it's transmitted through, through coughing, sneezing, contact with various different things, and is easily spread if it's part of the upper respiratory tract. So there's two phases to the disease. The first phase is when it's in the upper respiratory tract, where the epithelial cells are multi-layered, and therefore you might have a runny nose, you might have a lot of mucus secretion. And, especially in kids, if it's up there, they're sneezing, they're coughing. In a healthy person, a person who's sneezing and coughing, it's easy for the virus to be transmitted. However, if the virus continues to fester within the epithelial tissue, those cells are getting destroyed and eventually are going to lose motility. And the virus will then start to sink down into the trachea, the lungs, the bronchioles, and into the single cell layer of the alveoli, and really start to cause not just an immune response, but an inflammatory response. And as you can imagine, once someone is in this state, they're susceptible to upper respiratory disease where they can't cough the stuff up anymore, and then it leads to organ failure and all kinds of badness. So this is essentially what we decided to do. We're not going to show washing your hands. We're not going to show we know that it goes into the lungs. We went through many iterations, but essentially what we wanted to do was show this story through the eyes of a virologist, show what the structure is, the life cycle, and eventually some of the drug targets. This is just I get this question a lot, so I put this in here just to kind of answer the question of how I usually create a lot of my molecular visualizations. I use a lot of different software, but in this case, 95% of it was rendered in Adobe Illustrator, believe it or not. So this is the first part, the life cycle, where it comes into the cell It binds with ACE2, it comes in, the RNA is released, it takes over the machinery of the cell within the Golgi, within the replication complex where you find ribosomes, and it begins to transcribe and translate RNA, both in copying itself and also cleaving and copying and replicating and creating proteins that will eventually help in the process, but also build the virus itself. And eventually this cell is just full of virus, and through exocytosis, it's released and it starts the cycle all over again. But here are some of the 6 research targets that that Andy and I were discussing. So first, of course, there's monoclonal antibodies and convalescent plasma. So can we take some of this from patients that have recovered from the virus, from COVID, and use that as a defense? And then also we purposefully added chloroquine and hydroxychloroquine to the story. Now, before you start rolling your eyes and groaning, we did this purposely to say that they do not offer significant benefits. We really, really wanted to make sure that we were getting the correct information out there, that these particular drugs can be very harmful, especially when they're in combination with other things, can lead to heart arrhythmia and other heart issues. Of course, ACE2 inhibitors make sense. Can we stop it from binding? Inhibit protease activity? Can we stop these proteins from, working within the cell to produce other proteins and make new virus? Can we inhibit RNA polymerase and stop it from translating, transcribing? Here are a couple of visuals that kind of show some of the research that's going on right now from the Department of Defense. They're coming up with different drugs, and they're testing them. These are in clinical trials right now. And then last but not least, of course, a vaccine, which we're all so desperately waiting for. This is just to show what the final spread look like. So it shows the top, which is the structure, the middle section, the life cycle, and the bottom, the targets. And this is kind of I needed to create sort of a poster version because we were going to chop it up and use it in various different ways. And I am very aware of the time, so we're almost done. So here's the magazine, and here it is online, which if you go to this URL, you can see it. And then I'm just going to share very quickly some other visualizations from some of my colleagues. So if you want to see what the AMI is doing, what some of the people in our COVID group have been producing, on the AMI.org, if you scroll to the bottom, you'll see a lot of these different visualizations represented, and the list keeps growing as we speak. It just doesn't end. And of course, this spread was just one of about 15 or 16 projects that I've been working on. Here's a visualization of how the swab test is done in clinic. Most times we're doing the NMT procedure, but there is this nasopharyngeal one, which makes people cringe. And there's a video of that as well, so you can see that. And just recently, last week, in fact, I didn't even know there was self-swabbing until I had to do this rush job. So yes, there is self-swabbing. You can actually swab yourself and do this. So interesting. Again, shout out to Gloria. I have to say, this was one of the most helpful visualizations that came out of the group, because here, again, this is, I think this is probably Gloria's cheat sheet. Like, it has everything. And every time I was going to, I would put something down and I would guess what protein this is and blah, blah, blah. Like, wait a second, what did Gloria say? I wonder if I'm right. And I would check hers. I'm like, yep, that's what she says, the same thing. So I thank her so much, because this was so helpful. Of course, the CDC has been putting out some great visuals. If you ever go shopping, this should be on every wall of every store. And I apologize if David is watching. I had to turn your illustration sideways because I really wanted everyone to see the beautiful glory of this. This is a beautiful moment a sad moment, but a beautiful depiction of that moment of the replication cycle of the coronavirus. And you can see all of these on the Protein Data Bank website. They're all there for you to see in full color and full resolution. Kristen Brown at the National Institute of Allergy and Infectious Diseases has a 3D print exchange. And on the left, you can see her model, and on the right, you can see my printed model of that model. So that's an actual piece of plastic there. There's this beautiful depiction. On the right is a really nice illustrated depiction of how ACE2 binds to the spike protein. And the question is, and I've always had this question, I still don't know the answer. A lot of us don't know the answer. Is ACE2 really dimerized? And does it really need 2 spike proteins for this to happen? And according to Rami Amaro, I think it only needs 1. So who knows? So scientists are still guessing. There was a really exciting challenge on campus, the COVID-19 Design Challenge. And some of our graphic designers at Hopkins put together this nice flyer for people who are working in healthcare and are coming home, how to create a clean zone in your garage. Different visualizations about vaccine development timelines. We're even putting patient education material online for people who have diabetes because they're at high risk. There's a course at Hopkins you may have heard of. It's a MOOC that teaches people about contact tracing and how you can be a contact tracer. So these are some visuals from that. And journal articles. I mean, just various things that we're doing. So last but not least, I don't know, Sarah or Cappy, can you tell me, do we have time to actually watch this 2.5-minute video, or should I stop? Because I know I've gone over.
Jenny, I think that's fine. We are not terribly over. It's fine. Let's see it.
Okay, so let me switch over to my Chrome tab. This is the very end. So what I wanted to do is share with you, again, here's where you can find the magazine. So you can read all about and see the visuals that we created for the School of Public Health. You'll see all the targets here. But if you scroll to the bottom, there's a video.
What a virus wants to do is make more copies of itself and send it out. So coronaviruses are a rather large family of viruses. They are RNA viruses. Particularly in recent years, we've had viruses such as SARS-CoV and MERS, which are coronaviruses that have made the jump from another animal to humans, but have been associated with much more severe disease. And so SARS-CoV-2 represents the third of that, what I'll call the severe coronavirus diseases. Coronaviruses all have a very similar genome structure, but based on the specific sequences of their genes and the number of genes they have that are specialized for pathogenesis, you can differentiate one from the other. So the first thing a virus has to do is find a way to attach itself to a cell so that it can specifically get in there. That's the virus receptor interactions. For SARS-CoV-2, it binds to a protein called ACE2 that's on the surface of some, but not all cells. Viruses then have to get inside a cell. In the case of SARS coronavirus, it has sort of a membrane around it that hides its genetic material from the outside world. Your cells have the same kind of membrane around them that hides their material from the outside world. So when those two things come together, the virus has to find a way to get its genes inside the host cell. The virus genome, when it enters a cell, starts to find something in the host called a ribosome. And a ribosome is what turns RNA into proteins. It takes the sequence of the RNA and then it translates that into a protein sequence. RNA is primarily the means that we that express our genes. The more of the viral RNA that's in the cell, the more of that RNA will be translated into proteins, and the faster the virus can take over the cell. Their RNA serves those two purposes. It encodes the genes that it has to complete its life cycle, but it also is the way that it sends a message out to the cells to say, make more of my genes. And so within a few hours, there's a large portion of your proteins within your cell that are either from the virus or that the virus has somehow manipulated. And then towards the end of the virus life cycle, the virus sends out its proteins to particular places within the cell that are then the places where proteins are concentrated and new virus particles are made.
So that, by the way, was Andy's voice, which is what we wanted. We wanted the story to be told in his words, literally. So that was a lot of fun. Now let me just get to the end because I will be ready for questions in just a moment. Bear with me. Let's see here. Okay, so going to do this one more time. And okay, so that first slide that I showed you where we had 260 cases in the US, that was a snapshot that I had taken on March 6th. And on 2 days ago, on August 6th, I took another snapshot. And so as you know, these numbers are already out of, not accurate anymore. The cases keep growing. It's an interesting thing to look at. It's a sobering view. So this is where we were when we started, and now here's where we are today. And all I can say is, wear a mask, wash your hands, social distance, clean surfaces, be safe, stay home if you don't need to do what you need, if you can. Take care of each other, take care of yourself, and hopefully we will have a better story to share this time next year. I'm happy to share all the resources that I've collected through my research, and of course I want to acknowledge everyone in the Guild of Natural Science Illustrators. Thank you so much for having me. Thank you to the AMI and our group. Thank you so much to all the people that have helped me with this presentation. I hope I didn't miss anybody. And with that, I'm happy to take questions if we have time.
Thanks, Jenny. That was wonderful. I will pull up Sarah now so that she can run the Q&A. And then afterwards, we'll take a quick little interlude. Britt has a wonderful video that Scott Rollins made, the two of them together made, and we'll watch that video and then take a little break so people can get up and do what they need to do.
And Jenny, can you hear me all right?
I can, thank you.
Perfect. So I think we have time for one question.
Okay.
There's a lot of heat and excitement around your talk, and there are more than one questions. So again, I'll have Cappy, after you get done answering this question, I'd like Cappy to repeat what she said for anybody who's has joined since the first talk, so that people know how to access those questions and know that you'll be looking at all the questions and responding to them in the questions and answers section.
Absolutely.
Thank you. Thank you so much.
Happy to. Yeah.
So the question, the top question of the most burning question of the day for you is, at what point do you decide that you have enough information to complete the illustration and get it out there, even though research is still being developed? Particularly with molecular illustration, there are some details that just simply aren't fully understood.
Yeah.
So what responsibility do other illustrators and what responsibility do you have to be as accurate as possible versus creating imagery through beautiful representations that connect with audience? So
that is such a good question. I ask myself that every day. So there's a couple of things. Number one, of course, the thing that drives everything, for better or for worse, is a deadline, right? But that being said, in this, especially in this case, I mean, I still have questions. I'm not quite sure that I drew the M protein right. I don't, there, again, like, I don't know if this dimer, it doesn't, what does this do? So, and I will tell you that even if you go to the, the article page, you'll see the virus is like the second to last iteration that I had. You're not even seeing the other iterations I had before, which were wrong, but it was filled with RNA. And then I was like, no, it's not really like that. It's like this snake chain. So I drew the snake chain, and I knew that the RNA was also in connection with the actual proteins on the lipid surface. So so I was trying to draw it in such a way that kind of represented that for a lay audience. And then at the same time, I was like, no, but it's not hollow, it's packed, right? So when you look at the website, you'll see the hollow form, which is very stylized. And it's also, it's based that way because I want it to be like the simplified version. And that's the GNSI logo for this year. But really, that last image you saw of the RNA packed in there is probably more correct, but it's not, again, it's not 100%. So what we typically do as illustrators is when we report or publish something like this, we put a little line that says, this is the, for better or for worse, this is basically essentially created with the information that I had at the time in June of 2020. And even now, like, it continues to evolve. And then one day, another protein is going to come along that's put into the protein data bank or whatever. Yeah. And we're able to put that into and edit our illustrations. But of course, things go to press. And so thank goodness when they're in press, they have a date at the bottom of the page, so that's really the best we can do. Yeah, such a good question.
Well, I like your answer, which sometimes the simple solution is the best, which is just simply make certain that acknowledgement is there. This is an ever evolving story.
Yeah.
This is the snapshot in time for this date. So I like that. Thank you.
Thank you.
Well, this is kind of a mind-blowing talk, and we'll give everybody a you've been at the forefront. So Happy, I'll let you take over.
Yeah, it's a good segue to let people know that replays will be available at this exact site. You'll be able to access them through the end of August. So you will have plenty of time to come back and view this as many times as you need to. I also want to remind people that at the bottom of the window in your browser, you see the Ask a Question bin. We're asking Jenny to answer the questions. Each one, I believe she can comment on each one and answer them. So all of the questions will get answered. You can come back here at your leisure and read through and see if your question was answered.
Can I just to be sure? Can I then just click comment and answer them? Yes. And I can come back to this room anytime and do that, right?
Yes. Right. Yes.
Perfect. Okay, I will answer all your questions. I appreciate that.
Great. And so we're going to take just a couple minute break. Britt is setting up in the next session with a small interlude that we had. Scott Rollins created a small tutorial on how to create virtual field trips during this time to kind of break up online learning for students and other audiences. So stay tuned for that. And then afterwards, we'll have a longer break so that you can get up and take care of business if you need to before Quinn Burrell's talk. That will happen at 55 minutes after the hour. So we're just 5 minutes off schedule. Okay, so thank you, Jenny. That was a fantastic talk.
Thank you, everyone.
Thank you, everybody.
Okay, we'll see you all in Britt's session in just a moment.
Yeah.
Bye, everybody. Thanks for all the comments.